Hi everyone, welcome back to part 2 of chapter 22 on our lymphatic system. In part 1 of the lecture video, we ended with talking about the first 6 different types of ways our innate immunity is able to fight against pathogen non-specifically, and this is the last one we have left for the innate immune response. This one, they will sort of act as also as a way to alert us that something is wrong, and also as a way to slow down pathogen growth by triggering a rise in temperature on the body as a whole, which is kind of like what inflammation does, but instead of just raising the local temperature in that area where it's injured, it increases the whole entire systematic body temperature as a whole. And this is what we refer to as commonly as fever. Fevers usually can be triggered by chemicals called pyrogens that they can be released by bacteria, viruses, molds, yeast, and it also can be internally produced. Basically, if a fever happens, it really tells you something's going on in the body. Your immune system is actively doing what it needs to do to get rid of whatever that pathogen that has overtaken your body, not just at a local area, but maybe in other parts of the body as well. And a rise in body temperature can help with the slowdown on the growth of some viruses and bacteria, because like I said, some of those pathogens don't like, temperatures that are above 37 degrees Celsius. And more importantly, the increase in body temperature as a whole can speed up metabolism and enzymatic reaction that are working to repair the site of injury. 1 degree Celsius increase can increase 10% of metabolic rate. Okay, so that's a big difference in how fast things can work just by changing your body temperature by 1 degree Celsius, which is, the reason why when we get a low-grade fever, such as 38 or 38 degrees Celsius, we may feel kind of bad, but it's something we can handle. Sleep overnight, and you may be able to recover by the next day. Is because just by that 1 degree Celsius difference, you may already increase the ability of your immune system to do what it needs to do to get rid of the pathogen. And sometimes, of course, it may or may not be able to fight it, and you may, inquire or need interventions by other medications or medical procedures. Okay. So now on to our other way of fighting pathogens, which is adaptive immunity, a more powerful way to fight specific types of pathogens. So the adaptive immunity has to rely on the function of T and B lymphocytes, or what we call T cells and B cells. The mechanisms in which these two types of cells attack a pathogen are very different. T cells actually will differentiate into 4 different groups of cells for different purposes, as illustrated by this diagram here. The first 2, cytotoxic T cell and helper T cells, has to do with the actual immune response in fighting the pathogen. Cytotoxic T cells, as the name suggests, you can tell it's toxic to pathogens. So these cells will enter peripheral tissues and attack and destroy foreign cells or body cells that have been infected by the virus directly. Okay, so therefore that direct attack, cytotoxic T cells. Now, helper T cells is like the on switch for immune response. They don't attack the pathogen specifically, but they are absolutely vital to immune response, okay, or absolutely vital to our ability in fighting pathogens with our adaptive immunity. You need helper T cells to stimulate or turn on the activation of both T cells and B cells. And in the case of B cells, helper T cells must be there and working properly, of course, to stimulate the production of antibodies. So this is why helper T cells, even though they aren't really participating in the fighting process, but they are the on switch. And without the on switch, you can't do anything else with your adaptive immune response. Okay? And the third type of cells are called memory cells, and they are just as important because they are the very reason why after we get sick with a specific pathogen, the second time around that we see that same pathogen or become exposed to that same pathogen again, our immune system is able to fight a lot quicker and stronger that we don't even feel sick. Okay, so that's why memory cells are very critical for that part, that part of our adaptive immunity. And I'll talk about in detail why we need memory cells. There's also a fourth type of T cells called regulatory cells here, but we won't discuss about too much about them. Okay, so moving on. So cytotoxic T cells are the ones that utilize what's called cell-mediated immunity, okay? Or we can simply call it cellular immunity because they conduct direct cellular attack, as we have talked about just now. While B cells use what's called antibody-mediated immunity, or also can be called humoral immunity because they fight by production of antibodies that will bind to antigens on the foreign cells. Okay, they have to do with body fluids because humoral really means fluid, and antibody have to be released and distributed by your body fluids and bind to those antigens on the pathogens in order for them to be useful. And so what are antigens? We have already mentioned antigens when we're talking about blood typing, and we talked about them as something on the surface of the red blood cell. So technically speaking, they are just specific targets on pathogens or parts of the products of pathogen that can stimulate an immune response, okay? And these antigens are usually proteins, like protein receptors on top of a pathogen plasma membrane, or some lipids on there, polysaccharides, or even nucleic acids can serve as a way of antigens. This process of recognizing the antigen is called antigen presentation. Okay, so we won't talk too much about that process, but again, when you go to more advanced courses involved in immunology, you'll talk about the whole process of antigen presentation of MHC class 1, MHC class 2, but we won't talk about that here. You might get a little bit more from microbiology class as well in terms of immunity. Okay. All right. Once lymphocyte is able to make contact with an antigen, it becomes activated, okay, meaning that they will produce more copies of themselves so they can fight the pathogen with a whole army of cells. Just one or two cells is definitely usually not enough with the fight, especially when we're talking about adaptive immunity, which means somehow innate immunity was not able to stop this process, because innate immunity is usually the fastest to get to the site of infection. And why now the adaptive immunity have to step in to help with the fight is probably because the innate immunity is losing the battle. So they will need a lot of these copies of themselves. And those duplicate copies of adaptive immunity cells are called clones. And clonal selection is the process where we're looking for the lymphocyte that can recognize and bind to the specific antigen presented on that specific pathogen. Okay, allow me to go a little off into an analogy to explain this process. I've always explained this process to my students like a lock and key analogy. For example, the particular bacteria or virus that have taken hold in your body comes with a lock on their door, just as an analogy. And you have to open that door in order to know how to destroy or fight them. And as we know about lock and keys, that lock can only be opened by that one key that fits into the particular shape of the lock. So all the lymphocytes we have is like a huge pile of keys that we have in our hands. So in order to open that one lock for that particular specific virus and destroy them, we have to find that one right key within our immune system. But that pile of key, unfortunately, is huge. Well, fortunately and unfortunate both ways. There may be thousands and thousands of keys in that pile that we have to look for. So it's like picking one of those thousands of keys from the pile and trying to fit it into the lock. If it doesn't fit, we have to try another one. And if it doesn't fit again, we have to try the next one until you are able to find that one key that will fit into that one lock to open up that particular virus and be able to fight it. This is why sometimes we get sick for a whole week or even sometimes 2 weeks before we start to feel better from for example, like a flu. It takes time for our immune system to recognize that new foreign invader with a new lock we have never seen before. And thankfully, all that hard work in looking for the right key doesn't just get thrown out, okay? After we're done fighting with the virus, we have something like a little key box to identify that one key that will fit into the flu strain A and label them up, okay? We are able to have a memory of that identification. So with more exposure to different strains of whatever common cold or flu we will have a bank or little key box reservoir of identified keys for flu strain A, flu strain B, common cold A, etc. And this identification process is in form of our memory cells. Okay, that little key box is like our memory cells, so that when we are exposed to that same virus again, we don't have to go through the whole long process of looking for that one key in that pile of keys. That took forever. And we already know exactly which key it is and that is able to start the fight immediately to kill off that particular bacteria or virus. So this is why memory cells are very critical for our immune systems. And it also explains the very reason why vaccines work, okay? Vaccines usually involve injection of a dead or a weaker form of the pathogen into our body directly. That serve as a purpose to expose our body to that particular pathogen's lock or antigen. And it doesn't give us the full-blown sick symptoms like the original virus does, because it's either a dead version or a weakened version of it. So that way, our immune system can take their time to look for that right key without us having to suffer through what the virus does to our body. Once they're able to find that right key for that particular virus, they keep a memory of that of that correct match. And when we're actually become exposed to that original virus from the environment, our immune system already has a record of that and knows exactly which lymphocyte or the key will fit for that particular pathogen, okay? So, but if a virus has the capacity to change their locks very quickly, now that's in the case of, for example, like HIV, okay, that causes AIDS, then making a vaccine is pretty much useless, which explains why even till this day we don't have a vaccine against HIVs, because by the time we're able to produce a vaccine against that particular lock, the virus have already changed its lock within the individual, okay? So, which also, goes with the case of COVID-19 pandemic currently. I think research is still trying to figure out whether this particular virus for COVID-19 mutates quickly or not, or dramatically enough to see if a vaccine will be able to do its job. But of course, because of its urgency, we're still, we're already in the process of making the vaccine. But, in terms of whether they will mutate and cause the vaccine to become non-usable is still sort of not 100% confirmed yet. Let's put it that way. All right. So the whole story explains the mechanism in which our adaptive immunity is able to kill a pathogen, a specific pathogen. The antigen from the particular pathogen will trigger an immune response, which causes the phagocytes to be exposed to the antigen. This process activates T cells that are responsible for cell-mediated immunity. The T cell attacks the particular antigen, and also they will communicate back and forth between your B cells as well, where B cells can become activated via antibody mediated immunity, and they will mature into cells such as plasma cells that can produce a large amount of antibodies, and antibodies can help and bind and help with the attack of the specific antigen or the pathogen that produces those antigens, okay? Now, there are four types of adaptive immunity. We can categorize them into four different types, okay? One category is adaptive immunity that is called to be active, okay? Active adaptive immunity. It means that the individual themselves have to actually be exposed, okay? Exposure is the key word here, themselves, to the antigen, and the body responds by actively, okay? That's why it's called active immunity, because they actively make their own antibody to fight the particular pathogen, which is a consequence of our goal of our immune response. And this exposure of antigen can happen naturally by the individual encountering these antigens from the environment throughout their lifetime, okay? This is called naturally acquired active immunity, okay? So this is why oftentimes when a child is first sent to daycare, they may end up getting sick left and right because their immune system is what I mentioned earlier. They're still naive. Having, their immune system being at home all the time by you taking care of the baby and not really exposed to many people outside of their home, they haven't been exposed to a lot of pathogens at all. So once they, start playing around or are in touch with other people in daycare, then they can become exposed to all kinds of different pathogens, which is why they say, you need to let your child become exposed to different types of stuff, germs, okay, so that they can build their immunity. So this is what they're referring to when we're talking about naturally acquired active immunity, is allowing our immune system to become exposed so that our immune system can activate and start the process of our storage of our memory cells, so that when we become exposed to those pathogens again, we won't feel sick. Now, don't get me wrong about the situation with COVID-19, okay? There's too much unknown about that, what this virus does, okay? That the information about this virus is constantly changing. For example, in the beginning, when we first heard of COVID-19, they say they can't infect children, okay? So now they're saying they can infect people of all ages. Some become a deadly situation when you have COVID-19 infection, and some are mild and don't have any symptoms, okay? It seems to be just a very odd virus that scientists are still trying to figure out some of the patterns that it has, if it even has any. It's very weird. So, we stick with what the government says to do. Avoid contact with others. Don't be exposed just so you can acquire the immunity for COVID-19. We don't even know if one for sure can even survive through the attack. There are those that can survive and there are those that don't. And there seems to be some sort of pattern, but not really, because there's young people that die from the COVID-19 virus as well, okay? And the question of how long that acquired immunity, if it exists at all, can last for this virus is still unknown, okay? We don't even know we can even acquire the immunity for COVID-19. There's still some evidences for it, but it's still not 100% confirmed. Does being infected with COVID-19 means you won't get it a second time around? I don't think there is a 100% confirmed answer even till this day yet. So what I'm talking about here in terms of naturally acquired active immunity, we're talking about common pathogens that we already know a lot more about from years and years of experience and research about the certain disease. Not in this particular case of a rare virus that has caused this pandemic, okay? All right. The second type of active immunity is called artificially acquired active immunity, which, since it says artificial, it means that we don't encounter that particular pathogen from the environment naturally ourselves, but more so what I describe about what vaccines are doing, which is we're exposing the body in a controlled manner, okay, artificially controlled manner, where we know the virus is either dead or inactive, but it still serves the purpose of exposing our body to that, specific antigen from that particular virus. So that's why vaccines work, because it doesn't make us sick. Okay, we won't die from it, but it alerts our body, activates and makes those memory cells against that particular virus, so that when we actually become exposed to that particular virus, our immune system knows exactly which key fit into that lock to fight very quickly. Okay. Now, the other category of adaptive immunity is called passive immunity. And as its name suggests, it means it's something we get passively. Okay, we don't actively make the antibodies ourselves. So naturally acquired passive immunity is when we're giving someone else antibodies in a natural process. For example, when a baby is able to receive antibodies from the mom through the placenta, kind of like what we talked about in the blood chapter, when the baby is still inside a uterus, or through the breast milk after the baby is born, which is why they always encourage feeding the baby with breast milk at least for the first few months, to give them a bit of a boost of immunity from the mom, since when the baby is born, they have in a sense, a naive immune system, because they haven't been exposed to any germs or pathogens yet. Now, the other passive immunity is then artificially acquired passive immunity. This is when we are using human technology to isolate antibodies from one individual and transfer those to another person that need help with fighting an infection, because maybe their immune system is unable to, or they have a weaker immune system. For example, in the case of Ebola from 2014, the year of 2014, if you remember, it was a big scare back then too. When the person that survived Ebola gave blood plasma to the nurse, particularly in Dallas, Texas, there was 2 nurses that got infected with Ebola. She was able to recover from the infection after receiving that blood plasma. And that's a perfect example of artificially acquired passive immunity, because they have acquired those antibodies from the individual that have survived through the disease directly. Okay, but usually this is talking about short term. It's just giving a little bit more time for the immune system to act on their own and eventually help to develop that immune response. Okay, and why for Ebola's case is so critical is because Ebola acts way too fast for our immune system to have time to find that right key for that right lock. Okay, and before we're even able to maybe go through 10 keys, I'm just being analogous here. The virus itself already start attacking multiple parts of the body, and the person is already starting to die, which means their immune system is rendered non-functional. And that's actually where the Ebola virus first attacks, is the immune system themselves. So that's why in that particular case, artificially acquired passive immunity becomes a way to treat the individual. But for sure, they still have to be matched according to blood type in order to give this type of passive immunity. And here are some general properties of adaptive immunity that may be different from innate immunity. First of all, as we have discussed, adaptive immunity is very specific, which means they have specific defense activated by a specific antigen, where the immune response only target that particular antigen and no others, which explains why maybe 1 week you came down with a flu, okay, and you suffered through it. After 2 weeks, you become better. And then maybe the week after the next, you get sick again. And you're like, wait, I just got sick. Why am I getting sick again? Well, probably because you have contracted a different strain of flu. Your immune system might have memory of that particular flu strain A that you were exposed to the first time around, but the second time you were sick, you were exposed to flu strain B, okay, which means you start the whole process of looking for that right key for the right lock all over again, which is why you may get severely sick again a second time around. And also explains why flu shots usually contain up to 4 strains of common flu, is because we know that there are actually many types and strains of flu out there, and they sort of put the 4 most common strains of flu into flu shots as determined by World Health Organization, by statistics from them, and also they work together with your FDA as well to pick and sort of confirm which strain to put into the flu shot so that you minimize your chances of getting the flu, which is also explains why even after you get the flu shot, you may still get the flu, is because maybe that particular flu strain you got was not part of that 4 strain that was injected into you from the flu vaccine. It's not because the flu vaccine didn't work. It's because maybe that weird strain from another area has come and attacked our particular location, and you were hit with that particular strain that was not within the flu shot itself. Okay, so that explains how, in a nutshell, how adaptive immunity is specific for different pathogens. Okay. All right, and T cells and B cells usually respond by molecular structure of the antigen, by 3-dimensional shape and size of that particular antigen. And each, like I said, each one, or T cell or B cell, will bind only one specific type of antigen, okay? Which explains why it is only very specific to that specific pathogen. Now, versatility is another property of adaptive immunity, which means, technically speaking, our immune system is able to confront any antigen because we have a large diversity of lymphocytes and have a lot of variable structures of antibodies that can be synthesized. There are actually 1 trillion or more T cells and B cells, that can include millions of different lymphocyte populations. And within each population, they can contain several thousand cells with different membrane receptors. So the only problem is, in terms of why we still get sick, is because it depends on whether our body can handle and survive through the symptoms that the pathogen causes before our immune system can find that right key to fit in the pathogen's lock, okay, quote unquote. For example, like I said in Ebola's case, it was a tough fight because it can cause multiple organ failure very quickly by attacking our immune cells as one of the first thing they fight, okay? They somehow, I guess the virus in a sense knows, immune system is by adaptation and the evolution of them. They know immune system is what's going to cause their death. So the first thing they attack is your immune system. Before they're even allowed to look for the right key, they're being attacked already, which then allows the virus themselves to thrive and multiply very, very quickly and spread to other areas of the body very, very quickly, which was why Ebola was so deadly once a person was infected with the virus. And third property of adaptive immunity is memory, okay, which I talked about that immensely. Upon initial response to an antigen, usually 2 groups of cells are produced, one in which that actually kills and attacks the invader immediately. The other group kind of remain inactive as a memory, for when you meet that same pathogen for a second time around. Then there's a memory of them and you can fight very quickly. Memory cells are usually able to launch a much faster, stronger, and longer lasting attack that I'll talk about towards the end of the chapter once again. Okay. And lastly, a property of the adaptive immunity is called tolerance. Now, tolerance has to do with how our body won't attack our own cells with our own self antigens, meaning that, all cells, whether our own or foreign cells, they always have stuff on top of their plasma membrane that will identify them as what they are. Okay, it's kind of like carrying our driver license or identification card of some sort. Your cells have that identification card as well, but they're just in form of antigens. So there's security guards that are policing around your body, okay, and they look for these identification on you, okay. So if they see that, oh, you are our own cell, it's a self antigen, okay, I won't attack that one, you can move on and go on. But if they see a foreign invader with no identification, with a foreign identification, then they go ahead and, you know, sort of surrender them and sort of capture them and destroy them. So that's kind of like how our immune system is working and how it is able to prevent attacking our own cells, because what happens during T cell and B cell differentiation while they're being developed, any cell that react to self antigens are automatically destroyed by apoptosis. That's the process of, that's why they have need time to develop and need time for maturations. Part of that is they have to go through that process, that check, to make sure it doesn't attack our own cells, and that our own immune system should, in a normally speaking, react to foreign antigens only. Okay, so here is a brief summary of the mechanism in which T cells can carry out cell-mediated immunity. First of all, you have antigen recognition of the virus or the bacteria. Here's an infected cells that has virus in it, and here's a T cell that is able to recognize it by binding to its antigen. T cell then undergoes activation and cell division, mitotic cell division, to, you know, make your group of cytotoxic T cells and memory cells. The next thing it needs to do is those active cytotoxic T cell will destroy those antigen-bearing cell immediately on its own, directly. Okay, so that's why it's called cell-mediated adaptive immunities, because they destroy the target cells directly as a cytotoxic T cell. And the entire sequence, and as I've mentioned, of events from the appearance of the antigen to target destruction can take longer than what it sounds like just by words. Okay, usually 2 or more days, sometimes even weeks, may pass before the population of cytotoxic T cell reaches an effective level at the site of infection. So not only do they have to find the right lymphocyte that recognizes that specific pathogen, but they have to have time to multiply enough to be able to fight that particular pathogen. And thus, while we're waiting around for all that to happen, this can allow the infection to spread, and they can become more difficult to control after that time. So that's why I said it's not that our immune system is not capable of fighting certain pathogen, is the fact that can we wait that long and have a healthy enough of an immune system to develop that type of adaptive immunity we're talking about? Okay. All right. And here's a brief summary of the mechanism in which B cells carry out antibody-mediated immunity. It goes in 3 steps, just like the T cell-mediated immunity. It begins with sensitization, which is where the B cells finds the corresponding antigens. Okay, this is for example, antigen that's floating around in the blood, for example. And the particular B cells is able to find the corresponding one that can attack and bind to those antigen. And that's the whole process of sensitization. Then you have activation, where your helper T cell here is the action of your helper T cell. They will sort of, in a sense, they will have to okay or turn on the need for B cell activation, because after all, we don't want antibodies to be made out of the blue and start attacking things, anywhere they want. Okay, so in a sense, is used as a regulation and check and balance of your immune system to have these helper T cells around, which I'll later talk about, also serve as a breach in our immune system. If our helper T cells are the ones that's attacked by a pathogen, then it becomes the failure of response for our immune system. I'll talk about that when we get to diseases in the end. And last part of antibody-mediated adaptive immunity is division and differentiation. Your B cell would actually the one that actually recognizes the antigen, will then divide several times to divide into a little bit more daughter cells. And these daughter B cells will then differentiate into an army of plasma cells. Okay, plasma cells are the ones that's going to synthesize and secrete large amounts of these antibody in order to fight the pathogen. Remember what I said about by the time the pathogen is activating our adaptive immunity, that says something about the pathogen. It's pretty strong, and probably it has already multiplied many times into a big population. So that's why you need a lot of these antibody production very quickly, and that's why we utilize these plasma cells to make those large quantities of antibodies to be able to fight the pathogen. And at the same time, we have production of memory B cells, which currently is inactive until the second time around we are exposed to the same pathogen again. Okay. All right, so this is an animation video that goes over the process of these types of adaptive immune response that, sort of let you visualize what's going on. And it actually goes into a little bit about that, antigen presentation I was talking about with MHC class 1, MHC class 2 cells. But never mind that, you're not responsible for what I don't write on my PowerPoint slides. So, but it gives you a glimpse of what's actually happening for the immune system.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So that is an excellent animation that sums up that process we were talking about in terms of cell-mediated immunity and antibody-mediated immunity. So what about antibodies? How do they work specifically, and what are they made out of? So antibodies are soluble sort of Y-shaped proteins in general anyway. They can actually come in different shapes, but Y-shaped in general, and they consist of two pairs of polypeptide chain. One is called the heavy chain, which is this purple one right here, and one is called the light chain, which is this blue one right here. And each chain can contain both constant and variable segments. So you see how this is a solid purple let me get to my pointer. You see how this is a solid purple color. So solid purple color and solid blue color indicate that it is a constant segment, which means that it never changes between different types of antibody that is generated by different types of B cells or plasma cells. Okay, what changes is the variable segments, okay, which is highlighted by these slash, okay, or diagonal lines going through the purple, the heavy chain, or the blue, which is the light chain. Okay, so these variable segment is what enables different antibodies to be made against different specific pathogens antigen. Okay, that different three-dimensional structure in our antigen binding site is what then will bind to different antigens specifically. So constant segments of those heavy chain is what forms the base of the antibody molecule, and it is sort of the structure that determines the way an antibody is secreted and distributed. We're not going to go into too much details about that. And B cells actually produce 5 types of constant segments. There are 5 classes of different antibodies. Again, I told you immunologists love letters, so they are sort of given you by letters, IgG, IgE, IgD, IgM, and IgA. So they kind of come to form a word called MAG if you want to remember the different type of immunoglobulins or antibodies that B cells can generate. And of course, their constant segment or the base segment can be Y-shaped, and for some of them, they can actually be star-shaped, which is what IgM kind of looks like. It ends up looking kind of like a pentagon shape with stuff sticking out of it as the variable region. Okay, so depending what type, they can look different. I just didn't include that in here because we don't need to get into that much details about the specifics of these immunoglobulins, okay? Now, the specificity of an antibody depends on the amino acid sequence of that particular variable segments I was talking about up here, okay? Whether it be the heavy chain or the light chain. And the tips right here, these are the free tips of the antibody where they are called the antigen binding site, where different antibodies can be specifically made for a different antigen. And there are actually 100 million different types of antibodies that can be made, which is why I was talking about, technically speaking, our immune system is capable of recognizing any antigen out there. It's just whether we survive as a body, as a healthy body as a whole, before our immune system can recognize, okay, the specific antigen. So how the antibodies work once that there's one antibody that can bind to the pathogen's antigen is that they form what's called an antigen-antibody complex, where the antibodies must bind to the antigen on the cell just like this. For example, if this is a foreign invader, and there's those, or antigen itself, and there's these little yellow dots where your antibody is specifically able to bind to. These purple guys are the antibodies binding to those areas. And antibodies doesn't, wrap itself around the entire surface. It's only their antigen binding site, which is why you see that it's only that footy of the Y shape where that antigen binding site is located, that actually binds to the antigen-specific exposed region called the antigenic determining sites, or also called epitopes. So for example, if this was a particular antigen that either sits on top of a pathogen or is released by the pathogen, you see how the yellow, the antibodies is not binding to the entire pathogen, but they only bind to these little yellow spots on top of the antigen. And these little yellow spots are what we call the antigen determining sites or epitopes on that particular antigen, okay? and they can bind due to the three-dimensional structural fit, like enzyme's active site and its substrate. Kind of that same relationship, that three-dimensional binding that allows for that particular antibody to do its job. So here's a list of what this antigen-antibody complex can do. Although the antibody itself does not directly kill the particular antigen or the pathogen, it can enhance and also make the pathogen a lot easier to kill by other cells. For example, it can do neutralization, which means it is inhibition of the virus or toxin from binding to a body cell because it has rendered, sort of tied it down with antibodies. If you tie down with antibodies, that particular antigen have nowhere else to bind itself onto a body surface cell. I'll show you a quick animation video of this to illustrate this process in a bit. Now, precipitation and agglutination is what antigen-antibody complex can do, because if you imagine there's all these antibodies binding to the specific antigen, and it may create a three-dimensional structure called the immune complex. And if an antigen was, let's say, a soluble molecule, this immune complex can actually make it now soluble, meaning that it precipitates out of solution, which may make it easier for it to engulf and destroy. Now, if target antigen is on the surface of a cell or virus directly, for example, here's a bacteria and here's this antigen on there, and now all these antibodies are binding to it, binding to those antigens. So in a sense, it's like clumping those bacteria. So it does have a agglutination effect as well when we talk about how antibody can bind to these surface antigens on the pathogen. They can also attract phagocytes. An antigen being covered with antibodies is like an alert for the whole entire immune system to say, hey, something's wrong with this cell. We have just amplified that part for you, saying they're covered by antibodies. So you better attract more different types of eosinophils, neutrophils, macrophages to help with this particular fight, because now we have already identified them for you. You just need to come in and engulf it up. Okay? Opsonization has to do with the process of coating these antibodies so they can increase how likely they are to be phagocytized by those phagocytes. Okay, it kind of makes them easier to get a hold of. Okay, remember how we talked about the white blood cell needs to bind to receptors on the bacteria and form a vesicle around that particular bacteria, for example, and hence having antibodies sticking out of it kind of make that effect a lot more faster and also more effective for them to be bound by those white blood cells. So that's called opsonization. And then stimulation of inflammation too. When you have antibodies, some of them will stimulate your basophils and mast cells to release their histamine and do their job in causing inflammation and alerting your immune system that something is wrong. And lastly, they will prevent bacterial and viral adhesion. For example, the antibodies dissolve in saliva, mucus, and sweat is what make it difficult for those pathogens to attach to and penetrate our body surfaces, even though because those body surfaces are secreting these things, they have openings, but that's why we secrete all those things out of those openings to prevent those particular pathogens from trying to come into our cells. Okay. All right. So this is the video I was talking about. Before that, let me erase these so it doesn't cover it up. So it's a quick animation that goes over how having antibody complex is able to prevent the cell itself from the pathogen bacteria itself from attacking our cells. So here's the red blood cells, white blood cells floating in our blood vessels. White blood cells are those that looks a little more white. Here's a little single pathogen that has entered into our bloodstream with their little antigens, presented on top of their plasma membrane, and that's how they're able to bind to our cells specifically and starts infecting our cells. Well, let's say if a white blood cell was able to recognize that and bind to them, then they will produce antibodies, and these antibodies can bind to those specific receptors, rendering now not unable to bind to our cell. They can't infect our cells anymore, and our macrophages comes in to engulf them up and eat them up and destroy them. So that's how the whole idea of having an antibody antigen complex works. Okay. It's kind of like, I usually like to describe antibodies kind of like handcuffs, or those prisoner leg cuff that has a little big iron ball attached to it. Not little, but big iron ball attached to it to prevent the criminal from running away. So having antibodies attached to a pathogen's antigen is kind of like putting a hand or leg cuff on them, so that makes them easier to destroy and harder to run away. Okay. All right, so here's with the idea of memory cells, we can separate our immune antibody response into primary versus secondary response. So primary response is when it is our initial or first time exposure to the particular type of antigen, and it will take time to develop because we talked about the antigen must activate those appropriate particular B cells, which needs time to differentiate into plasma cells, and they need time to produce antibodies. So the antibody titer, which refers to the amount of antibodies within the plasma, antibody titer, actually does not peak until 1 to 2 weeks after initial exposure. Again, explaining why we can be sick for so long before we start to feel better is because that's how long it takes before our antibody is able to peak in concentration, so they can help with the immune response. And if we're no longer exposed to the antigen, the antibody titer usually will decline. But again, like we talked about, we do have those memory cells. That's why we make those memory cells for a second time exposure, so we can fight very easily. Okay, plasma cell, contrary to memory cells, usually have a short lifespan. So once the antigens are gone, okay, the pathogen is eradicated, the plasma cell will then die off, and we no longer are making those antibodies until our memory cells are activated again during a second exposure. And hence the IgM here, okay, the book does make a distinguish between IgM and IgG, and I want to talk about this is because there is a difference in terms of how quickly each respond and which one ends up being the one that is responsible for killing off the pathogen. IgM is usually the first antibody to fight against pathogens. But IgG is actually the one that can cause or help with the permanent eradication of the pathogen. They are slower to react, but they are more powerful, and they can enhance phagocytosis or opsonization that we talked about, and it does so for a longer period of time compared to IgM. So eventually, we're really relying on the production of IgG to help and finish up the fight. And hence, come second time the body is exposed to the same pathogen, we call it a secondary response, because now memory B cells are already in place for that specific pathogen. And they will initiate an immediate response due to the quick activation of those B cells. Now they start producing huge amounts of IgG very quickly, and which I was talking about, IgG are more effective and destructive type of antibodies that are produced to kill off the pathogen or the antigen before you even realize you're sick. So sometimes maybe we were exposed to the second time of that particular flu strain, but we just never felt sick, is because our immune system was able to react in such a quick manner when is the second time around. Now, and so sometimes this secondary response still appear years after your primary response, which is why it is critical for these memory cells to survive a long time, sometimes even more than 20 years, which explains why vaccines that we are given when we are babies or as a child is something that we may not need a second shot of later in adult life, is because once we have a memory of it, that particular memory cells can survive a long time. Okay. All right. So this graph here gives you a summary of the timing and the amount in which innate and adaptive immunity can work together to help us fend off pathogens as a general scheme. Okay. So during early stages of infection, it is the neutrophils and natural killer cells very quickly that acts very fast and migrate to the infected area and try to destroy whatever it is, bacteria, virus, etc. And if that particular things, a pathogen still survives, macrophages then come help with the fight because neutrophils will release stuff that recruits the macrophages. And at the same time, if your adaptive immunity is able to recognize that particular antigen or pathogen, cytotoxic T cells will start to become activated and becomes high in amounts, which then leads to your B cell activation because they communicate with each other. Your cellular mediated immunity versus your antibody mediated immunity. And once your B cells are activated, then they will give rise to a bunch of plasma cells, as indicated by this blue line here, which you can see that it takes almost 1.5 weeks to peak in population. Then it takes some time to create that, large population of antibodies that is generated by your plasma cells. So your antibody level, that's why it doesn't really actually peak until 2 or 3 weeks after from the time of initial exposure to the pathogen. So it depends how strong that pathogen is. Sometimes they're killed off immediately in this innate response, by our innate response of natural killer cells and neutrophils and macrophages. But if they survive past this stage, then we really got to rely on our adaptive immunity to do its job. And the last thing is try to use those antibodies to help render and, surround these pathogens and kill them off. Okay. So that's why immune depends what stage it is and what type of pathogen. It can take longer time. All right, and lastly, as a last sort of concept we'll talk about to wrap up this chapter are immune system diseases. Okay, as much as our immune system is supposed to be powerful and help us fend off a lot of pathogens, there can be times or conditions that leads to it being either ineffective or even having adverse effects. For example, hypersensitivities is the case where the immune system leads to negative adverse effects. Sometimes, somehow, the immune system can generate an excessive amount of immune response to certain antigens. For example, neutrophils or cytotoxic T cells are supposed to destroy only just the antigen, but somehow they start destroying normal cells while attacking the antigen, or the antigen antibody complex may trigger massive inflammation instead of just at that area. Okay, or in the case of allergies, where one can be allergic to pollen or dust, or other stuff, or nuts, for example. Those aren't really pathogens per se, nor do they harm our cells, but somehow our immune system recognizes them as a potential harm and starts activating all the responses that it doesn't need to. Pollen is not going to kill us, but that's what some of us can become allergic to. And in some cases, an anaphylaxis is what can result from a severe allergic reaction where a range of symptoms can occur, including swelling, edema, hives, or even constrictions of your airway that can limit your ability to breathe and suffocate you. Okay, so in severe cases, extensive peripheral vasodilation can also occur. Other than constricting your airways, you can end up with a quick drop in blood pressure, and if it's low enough, okay, we can, as we learn from our blood vessel chapter, low blood pressure results from vasodilation. And if it is low enough, it can lead to what's called circulatory collapse that we talked about. And it actually can, cause the individual to go into what's called anaphylactic shock, which is, stopping of the heart because there's not enough blood that can go into your brain and your heart at this, because due to that super low blood pressure, stimulated and regulated by your, stimulated by your immune system in a sense. Okay, so usually antihistamine is usually what's given to block the action of these hypersensitivities, and there's a whole variety of different antihistamines out there depending on what works for each different individuals. Now, the other thing that can happen to the immune system is sometimes out of the blue, the immune system can attack the body's own healthy cells. We call these autoimmune disorder because they attack our own normal cells, and somehow, and till this day, it is still not very well understood why this happens. That's why I said it happens out of the blue, because that's exactly what some of those autoimmune diseases do. You were just your normal self for the longest time. You didn't change much in your health, diet, or your exercise routine or anything else. You don't take drugs, you're not smoking, but somehow an autoimmune disorder kicks in. A lot of times that's what happens with these autoimmune diseases. And somehow we just know that those antibodies became misguided and they start attacking specific areas or tissues of the body when they are perfectly normal. For example, we talk about rheumatoid arthritis last semester in the joints chapter. Where the immune system will attack the joints specifically. Okay, why it does that, we don't know. Or in the case of type 1 diabetes, where type 1 and type 2 diabetes, how they differ is type 1, there's an early onset of diabetes, usually starting from when they are at a young age, whereas this type 2 is due to usually happens at a later stage and has to do with your environment and dietary routines and habits as well. But of course, there's a genetic portion of type 2 as well, but type 1 is definitely something that is triggered in early stages because it stems from antibodies that attack the pancreatic islet specifically. Okay, so because it's a malfunction of the very complex immune system itself, a lot of times autoimmune diseases are very tough to find a permanent cure to, and in a lot of cases we can only manage the level of response sometimes by inhibiting or turning down the specific pathway of the immune system if the pathway can actually be well defined for that particular autoimmune disease. Okay, for example, if let's say this particular autoimmune disease has to do with T cell activation, then they may give you a drug that that can decrease the amount of T cell response. But of course, you compromise that for your normal immune response as well when you are in those type of therapy. Okay, and there are also cases where one does not have an immune system at all. Okay, and this can happen naturally or from a viral infection like HIV. Well, SCID is a natural process that is due to genetic errors. Okay, it stands for severe combined immunodeficiency disease. Immunodeficiency itself tells you that they don't have an immune system. Usually these are happening in babies, and they are born with a failure to develop either cell-mediated or antibody-mediated immunity. Usually they have very low lymphocyte population or complete absence of B cells and T cells within their immune system. And this makes them very much susceptible to any opportunistic disease or any small amount of germs that we can easily recover from. They can die. And that's why if SCID babies were not treated without any treatment, very minimal infection can actually lead to death. And infants with SCID usually die from an infection within the first or 2 years of their life if they don't have any treatment at all. And unfortunately, because it's an autoimmune disease, the most effective treatment for SCID, and they can be actually cured from it, is a bone marrow transplant. Okay, so remember, red bone marrow is where we have all these hematopoietic stem cells, so they can acquire new hematopoietic stem cells from a bone marrow transplant, and that allows them to generate those immune cells properly again. So which is why if they are able to find the right donor, they can potentially recover fully from SCID once they have a bone marrow transplant. Well, the difficult part is finding the correct match donor is usually takes a while. Okay, and so you only have that small window before the child can or baby can pass away from the effects of SCID. Now, the other immunodeficient disease as a result of virus infection is HIV infection. Okay, so, well, HIV is a type of virus that targets specifically your helper T cells. Remember I said that helper T cells is very vital to the function of your immune system properly. Okay, if you don't have helper T cells, you can have as much T cells and B cells in your immune system, but they're useless. Okay, because you are not allowing helper T cells. There's no helper T cells to turn on the activation of cloning this group of particular T cells that can attack specific pathogens. Like I said, having just one pathogen, one immune cell that can bind to that specific pathogen is not enough. You need a whole army of them. And without helper T cells, you cannot make those clonal groups, okay? And you cannot make multiple soldiers or groups of things that can help fight with the battle or pathogen at the same time. So without helper T cells, it's pretty much a non-functional immune system, which is why at the end with full-blown AIDS, people die from opportunistic diseases, okay? Because they don't have an immune system, which renders them close to the case of what happens with SCID patients. All right, and that's all I have for you. It's a pretty short chapter compared to the other chapter. Let's go over the practice question and we'll be done. All right, so which of the following is not a function of the lymphatic system? Very easy. They don't drain blood into the right atrium. That is for the job of the cardiovascular system. Immune system or your lymphatics will drain blood into your left and right subclavian vein, but not specifically your right atrium. Which of the following is true of lymphatic capillaries? They have endothelial cells in the capillaries, lymphatic capillaries, that are not bound tightly together, but they do overlap to form valves, okay, as we've mentioned. Which of the following is a dangerous consequence of lymphedema in the limb? It can accumulate toxins and pathogens that can lead to stagnant interstitial fluids and also chronic infection around that area. If the thymus failed to produce thymic hormones, which population of lymphocytes will be affected? Of course, your T lymphocytes, T thymic for T lymphocytes. The spleen is both A and B. It's a fragile organ that can rupture easily, and it does contain red blood cells within the red pulp. And the white pulp is where lymphocytes are located. A tumor cell has developed and begun to divide in Amy's liver. What type of immune cells will be able to detect and kill tumor cells? Remember, our almighty natural killer cells that are able to fight a vast variety of different types of cells. What effects do pyrogens have in the body? Pyrogens is for fever, so they increase temperature of the body. A decrease in the number of cytotoxic T cells directly affect which type of immunity? Cytotoxic T cell. They're toxics directly, so it's cell-mediated immunity, okay, because they kill the cell directly. How does the absence of helper T cells affect the antibody-mediated immune response? Neither B cells or T cells are stimulated to respond to a pathogen, so no immune response, which is why I said helper T cells is absolutely vital to the function of your immune system. Which kind of immunity protects a developing fetus, and how is that immunity produced? Is it natural? Well, first of all, is it active or passive? Okay, we're talking about helping a developing fetus, so it's passive. And is it naturally or artificially made? Natural, because this is natural antibodies developed by the mom that crosses over through the placenta. Okay. Artificially acquired passive immunity is blank, while artificially acquired active immunity is blank. Well, passive means it is, given by, administration of human technology, which is given you straight antibodies from someone else through IV. Whereas the other one, you're talking about active. So it causes the body to actively make antibodies themselves after the actual exposure to an antigen. Okay. Which of the following is not a characteristics or function of non-specific defenses? Dependence on activity in lymphocytes. Lymphocytes are only for adaptive immunity or specific type of immunity. Okay, so all these other ones are characteristic of innate response. How is the secondary response affected if memory B cells for a particular antigen are absent? The secondary response won't occur. Okay, very plainly put, because memory cells is solely for this secondary response. So if we don't make any memory cells, we have to take 1 week, 2 weeks, 3 weeks, however long it takes to find the right key all over again if we don't have memory cells. Okay, and that's all I have. See you in the next chapter.